in the cell cycle, we talked about how the cell progresses unidirectionally around the cell cycle in only one direction, and it somehow knows when it's ready to go to the next phase. And throughout mitosis, things happen sequentially and in the correct order, basically automatically. If we look at the cell cycle, we as humans have divided this up functionally. The G1 phase, the S phase, the G2 phase, then the M phase. And the M phase has been broken up into 5 distinct regions, whereby in the middle, right before anaphase begins, there's a checkpoint that occurs. It's a checkpoint. We, as humans, put this in a circle, and we say, OK, this is the cell cycle. It progresses in this order and in this fashion. And we know that it does. And it makes sense, because in the G1 phase, this is where the cell needs to respond to proliferation signals, decide if it should go into the cell cycle or not. It's also where it needs to then initiate the replication machinery that's going to be required in the S phase, and perform DNA checks to ensure that this is safe to do. And we can say, well, if the G1 phase has all of its boxes checked, it's ready to move to the next level. Move to the next level. And it does. It passes a checkpoint and moves into the S phase. And the S phase had a purpose. The S phase is replication. And in the S phase, DNA gets replicated. And before it leaves the S phase, it ensures that the DNA nuclear material went from 2N in G1 to exactly 4N at the end of S. Right? The DNA had to be doubled, exactly, all the DNA replicated, but all of it only replicated once. And it has to assess for problems. Was DNA repair needed? And as soon as all of DNA is replicated, it can turn off the cellular machinery for its synthesis and move into the G2 phase. And the G2 phase has a purpose. The G2 phase was to increase the amount of cytoplasm, increase the number of organelles, and do one last DNA check. And if everything checked out, well, then it would move from G2 to M. This makes sense, right? We want to dedicate cellular machinery to doing what each phase needs to do, and should progress in this order. How does the cell know to do that? It's not thinking logically, but yet the cell cycle does occur clockwise, unidirectionally, and the cell won't progress to the next phase unless all the boxes are checked, unless it's completed the task for that phase. Well, how does it do that? Well, there's a number of ways it happens, but in researching this, what we discovered first was this mystery protein called maturation promoting factor. And maturation promoting factor allowed the cell cycle to progress from G2 into M. Well, it turns out what MPF is simply the combination of a protein called a cyclin with its corresponding cyclin-dependent kinase. Now, cyclin-dependent kinases don't do anything when they're not bound to their cyclin. But you put the matching cyclin to a cyclin-dependent kinase, and what you get is an activated CDK with kinase activity. In this case, it was a combination of M-phase cyclin, M-cyclin, and a CDK that was MPF. Now remember, moving from G2 to M phase sees the nuclear envelope break down, the DNA condense in the mitotic spindle form. How does it know to do that? Well, it turns out that M-cyclin, together with its CDK, has a kinase activity, cyclin-dependent kinases. The kinase activity is turned on by binding its cyclin. Well, the phosphorylation that the kinase does leads to the nuclear envelope being degraded. It leads to DNA being condensed. And it leads to other transcription factors and phosphorylating proteins cytoplasmically and nuclearly that allow all of the process of mitosis to occur. And mitosis always lasts about the same duration, so always about 1 hour in an adult cell. That's because in addition to activation or inactivation of downstream targets by phosphorylation, the cyclin CDK complex also degrades cyclin. It's a cyclinase activity, which therefore results in an inactive cyclin-dependent kinase. Over time, cyclin CDK complexes dissociate as cyclin is degraded. It's therefore the expression of the cyclins that are going to allow the cell to activate MPF, which then phosphorylates downstream targets that allows the cell cycle to progress. Okay? MPF was a mystery word, so I'm going to use MPF for the combination of cyclin and CDK. In G1, M-cyclin isn't made. It's D-cyclin. In G1, cyclin is expressed. D-cyclin is expressed. And when cyclin D combines with the CDK, it has a kinase activity. What cyclin D does when bound to CDK is it tells the checkpoint signal, the guardian of the checkpoint, that it's OK to move forward. It's retinoblastoma. We'll explain that in a second. But in addition to passing the checkpoint, that kinase activity also activates S-phase genes. And one of those S-phase genes is to make the S-phase dependent cyclin. Now, I've used M-cyclin and S-phase cyclin to mean mitosis phase cyclin and S-phase cyclin. I don't want you to learn the different cyclin letters that correspond to the different phases. I do want you to learn cyclin D, which is the letter D for the G1 cyclin. G1 cyclin, cyclin D is what it's actually called. I don't want you to learn the S-phase or M-phase cyclin names. Just learn cyclin D. But the point is that in addition to passing the checkpoint, it also activates the genes of S phase, which include the S phase cyclins and the S phase machinery. And because the cyclin CDK complex has a propensity to degrade the cyclins, the progression in one direction is maintained. See what that looks like graphically. I want to compare the level of cyclins to the CDK activity. Cyclin-dependent kinases are constitutively expressed. They're always at the same level, but their activity changes and varies with each cell cycle phase. And not surprisingly, you'll see an activation of the G1 phase CDK activity during G1 and the S phase, the G2 phase, and eventually M phase. Now, I am not drawing these strictly. There actually have a lot more crossover than I'm implying, but it's to illustrate the point. Cyclin-dependent kinases have variable activity because of variable cyclin expressions. Cyclin D, the G1 cyclin, is expressed in exactly the same pattern as the cyclin-dependent kinase activity is found. And the same is true for the S phase, G2 phase, and M phase. So as the cyclin levels specific to that phase increase, cyclin-dependent kinase activity specific for that phase increases as well. It's the combination of the cyclin with the CDKs that increases the CDK activity. Since cyclin-dependent kinases are constitutively on, it's only the variable expression of cyclins. The genes that code for the cyclins are activated or inactivated at various time points during the cell cycle. The way the cell ensures unidirectionality is by having the cyclin CDK complex initiate the transcription of the cyclin for the next phase, so that the G1 phase cyclin turns on expression of the S phase, and only the G1 cyclin CDK combo turns on the cyclin for the S phase. S phase for G2, G2 for M, and so on. Cyclin levels rise by gene expression. Cyclin levels fall by the innate degradation of the cyclin. And the falling apart of the cyclin CDK turns off the function of the kinase, which also had kinase targets specific to that point in the cell cycle. Okay, cyclins turn off by time. They get degraded. They get turned on by gene regulation. Turning them on activates CDKs, progresses the cell cycle forward, and turns on expression of the genes for the next phase. What then controls the cyclin? Because we know that if there's cyclin activity, that is, the cyclin gene is expressed, it will activate the CDKs, and the cell cycle will progress. On one side of the genome are genes that are known as proto-oncogenes. Activity of proto-oncogenes is pro-proliferation. These are signals that are looking for positive signals. These things must be positive in order for cyclins to be expressed. It's all about a balance of cyclin expression. Yes, the previous phase's cyclin CDK complex initiates the transcription of that phase's cyclins, but it's not just that signal. The proto-oncogenes exist as a sequence, such that the product of one gene gets activated and sends a signal back to feed forward down the sequence of events that ultimately results in a transcription factor that activates the cyclin gene. This cascade ensures that multiple positive signals are received in order to activate the cyclin gene. Once the cyclin gene is expressed, the CDK activity goes up, the cell progresses through the cell cycle. Proto-oncogenes are named proto-oncogenes because if they're mutated and cause cancer, they would have a gain of function. That is, an overactive gene doesn't wait for its upstream activation, it just pushes the cell towards proliferation, so it loses some sort of regulation. Proto-oncogenes are pro-proliferation, and a single gene gain of function can lead to unregulated proliferation. On the other side are tumor suppressor genes. Tumor suppressor genes do not act in sequence or cascade. These are the cell cycle checkpoint regulators. They're also the things that check for problems. They are anti-proliferation. And what the tumor suppressors do is generally look for problems. Because their positive activity results in the arrest of the cell cycle, mutations that would result in unregulated proliferation require a loss of function. Generally, that means both genes need to be knocked out. Proto-oncogenes move forward, tumor suppressors inhibit. All right, what's the most well-known example of this system is that G1 checkpoint. Cyclin D combines with its CDK. When it does, it activates the kinase activity of CDK, which phosphorylates something. The target of that phosphorylation, as we talked about already, is going to be the S-phase machinery, the S-phase cyclins, but directly, the cyclin D CDK combo phosphorylates the protein retinoblastoma. Normally, retinoblastoma is sequestering a transcription factor, E2F. And with the kinase activity, phosphorylation of retinoblastoma
أصغر كاميرا مراقبة بجودة عالية لمراقبة مكان العمل والبيت أو أي مكان تريد مراقبته عن طريق هاتفك وبشكل مباشر. تتوفر على
In the cell cycle, we talked about how the cell progresses unidirectionally around the cell cycle in only one direction, and it somehow knows when it's ready to go to the gene. Once the cyclin gene is expressed, the CDK activity goes up, the cell progresses through the cell cycle. Proto-oncogenes are named proto-oncogenes because if they're mutated and cause cancer, they would have a gain of function. That is, an overactive gene doesn't wait for its upstream activation, it just pushes the cell towards proliferation, so it loses some sort of regulation. Proto-oncogenes are pro-proliferation, and a single gene gain of function can lead to unregulated proliferation. On the other side are tumor suppressor genes. Tumor suppressor genes do not act in sequence or cascade. These are the cell cycle checkpoint regulators. They're also the things that check for problems. They are anti-proliferation. And what the tumor suppressors do is generally look for problems. Because their positive activity results in the arrest of the cell cycle, mutations that would result in unregulated proliferation require a loss of function. Generally, that means both genes need to be knocked out. Proto-oncogenes move forward, tumor suppressors inhibit. All right, what's the most well-known example of this system is that G1 checkpoint. Cyclin D combines with its CDK. When it does, it activates the kinase activity of CDK, which phosphorylates something. The target of that phosphorylation, as we talked about already, is going to be the S-phase machinery, the S-phase cyclins, but directly, the cyclin D CDK combo phosphorylates the protein retinoblastoma. Normally, retinoblastoma is sequestering a transcription factor, E2F. And with the kinase activity, phosphorylation of retinoblastoma causes a conformational change that releases E2F. When retinoblastoma binds E2F, the promoter for the E2F gene is not bound, and there is no expression of that gene. If retinoblastoma gets phosphorylated by cyclin D CDK, E2F is released. E2F can bind to the promoter and express that gene. That gene leads to movement from G1 into S. Cyclin D, proto-oncogene. Retinoblastoma, tumor suppressor. In apoptosis, we talked about another tumor suppressor called P53. This is going to be used to illustrate a couple of points. When there was DNA damage, P53 was phosphorylated, thereby being released from the protein MDM. Phosphorylation of retinoblastoma allows cell cycle to progress. Phosphorylation of retinoblastoma is inhibitory to retinoblastoma, but pro-proliferation. Phosphorylation of P53 activates P53 and leads to cell cycle arrest. Now remember, in the context of apoptosis, we said low affinity receptors are activated by increased concentrations, which lead to apoptosis. That was the Bax gene, Puma nova. We said something about some moderate stuff that leads to senescence. I don't want you to learn that here. But we also just kind of vaguely said, in low concentrations, P53 activates high affinity receptors that induces cell cycle arrest and turns on DNA repair. Well, what does induces cell cycle arrest mean? P53, when activated by phosphorylation, is a transcription factor that binds to DNA at high affinity receptors to produce cyclin-dependent kinase inhibitors. Cyclin-dependent kinase inhibitors bind to the cyclin D CDK complex and inhibit the kinase activity. So the cell progresses unidirectionally clockwise around the cycle. Each phase is dedicated to doing something specific. G1 is deciding whether or not it should divide, preparing for replication. S phase is replication. G2 is getting bigger so that it can divide. M phase is about separating to two cells. It maintains unidirectionality because the cyclin CDK complex both pushes the cell through the checkpoint and activates the next phase's cyclins, such that only the S phase cyclins turn on only G2 phase, so the cycle must progress. Checkpoints exist along the way to ensure the cell has done what it's supposed to do. At G1, it's the retinoblastoma gene, which is phosphorylated and deactivated by cyclin D CDK levels. Cyclin D, the cyclin gene, is expressed by the activation of proto-oncogenes in the absence of tumor suppressors. At the G2 phase, we had that MPF, which was simply M-cyclin binding to CDK. Once this happens, mitosis starts, and the inherent activity of the cyclin CDK combination dissociates, cyclins degrade, and the process is reversed. leading to 2 daughter cells with a reinitiated nuclear envelope and non-condensed DNA. In metaphase, there is another checkpoint which we're not going to explore here, but that ensures all of the chromosomes are at the metaphase plate. Tumors can be generated by increasing proliferation inappropriately, by activating proto-oncogenes, or by losing tumor suppressors. Cell cycle regulation is about the cyclin levels, cyclin phase-specific expression, and cell cycle phase-specific cyclin-dependent kinase activity. Activation of proto-oncogenes moves forward, activation of tumor suppressor genes slows it down. That is cell cycle regulation.